Thank you very much for kind introduction and invitation. I'm very keen to be here to learn chemistry. I'm not a chemist, so my talk will be very different to what you heard before. There will be no click chemistry. There will be a little bit of chemistry, but what we study is what chemistry happens to DNA when it's in cells. And particularly at the moment, one of the projects which I'm going to talk to you about is interested how mutations emerge in DNA. And mutations, when they emerge in the DNA, they are problem which leads to cancer, and this is what we're studying, like the inputs for cancer research. Okay, so 4 bases in the DNA, one of them is unusual. It's unusual in vertebrates because it can be modified in the DNA. And 2 major modifications which are observed are 5-methylcytosine and 5-hydroxymethylcytosine, which can be found from around 5% to 2.1% in the genomes. And there are other 2 minor modifications, 4-methyl-carboxyl-cytosine, and 4-methyl-cytosine was identified with Thomas Carell. And so Zhang, and these are modifications which are very rare in the genome, but they are serve very important functions. So how DNA methylation patterns, so now focusing on 5-methylcytosine, look like in the genome. So first of all, methylated cytosines we find in CpG dinucleotide context, making them kind of symmetrical around both strands. So within the CpG dinucleotides, and if we look at the distribution of methyl CpGs in the genome, we see that there are proportion of methyl CpGs which are highly methylated, and there's a small proportion which are poorly methylated or unmethylated. And if we just zoom across model gene, the methylation patterns look like this. So majority of the genome is methylated. That's why we have high proportion of methylated CpGs. But places where we observe unmethylated CpGs are CpG islands and typically enhancers. So these places which have slightly higher density of CpGs than genome average, and they are unmethylated. So 5-methylcytosine, when CpG islands get methylated, is associated with repression of transcription. And this happens in some circumstances. Typically, CpG islands are methylated in most of the cells, but they can get methylated. So some genes in cancer cells can acquire DNA methylation and be silenced, and this is a phenotype known CpG island hypermethylation phenotype. Some genes which are expressed in testes only and repressed in the rest of the body as well, maintained by DNA methylation silencing. Imprinted genes, so these are genes which differentially express whether you inherit them from your father or from your mother, and one of the alleles is being typically silenced by DNA methylation. And in females, 2 X chromosomes, and due to dosage compensation, only 1 X chromosome is active, and the inactive X chromosome contains hypermethylated CpG islands. So these are circumstances where methylation can happen on CpG islands and with silent genes. And so in the majority of the genome, we have lots of non-coding retrotransposon, repeat elements and satellite repeats, and they are maintained methylated as well. So how DNA methylation is deposited? We have in humans 3 DNA methyltransferases, DNMT1, 3A, and 3B, and this would methylate cytosine within the CpG nucleotide. So 3A and 3B are genome methyltransferases. They're important to deposit DNA methylation when cells, for example, change their identity, and DNMT1 is a maintenance methyltransferase. So it's very important when cell divides to maintain methylation patterns on the daughter strand after replication. So I'll introduce other enzymes. So these are TET123 enzymes. So the name comes from 1011 translocation. So they were first observed being translocated in cancer before the molecular function was characterized by Antanas Rao group. And these are oxygenases which will use 5-methylcytosine as a substrate and will create hydroxymethylcytosine, and using iterative oxidation step will create 4-methyl-carboxyl cytosine. So hydroxymethyl cytosine is quite stable base, so we observe a lot of it in the genome of brain cells, and we don't know exactly why. There are some hypotheses, we don't know exactly why brain is so unique and enriched in hydroxymethyl cytosine, but formal carboxyl cytosine are very rare in the genome. So this iterative oxidation is important for removal of methylated cytosine. So one way it can be removed is essentially by having, for example, carboxyl cytosine, and when DNA is replicated, one DNA daughter would contain a carboxyl cytosine, another would have unmethylated cytosine, and carboxyl cytosine, hemimethylated, hemicarboxylated sites are very poor substrate for DNA methyltransferase maintenance. And there is an active mechanism which probably operates in non-dividing cells where carboxyl cytosine is excised by finding DNA glycosylase, creating a basic site which then is cleaved, and using short patch repair or long patch repair, it can be repaired by the synthesis of the strand, leading to incorporation of a modified base instead of modified base. So DNA methylation is essential for some organisms, but it's not universal in life. So, for example, if we take out DNA methylation completely from cells in mice, it will not be compatible with normal differentiation and development. But some organisms don't have DNA methylation, a lot of DNA methylation, and actually these are very frequently model organisms which biologists work with. For example, yeast, insects like flies, drosophila, mosquitoes, nematodes don't have at all DNA methylation. So it's not that essential to life as a whole. So it's quite interesting that the biological DNA modifications kind of have intimate link with DNA damage. And I'll give you a few quite interesting examples. So one, I showed you that to remove carboxyl cytosines and remove methylated cytosines, you employ some of the proteins which are involved in DNA damage repair. But as well, the enzymes which cause the DNA modifications, they have what they call rogue activity. So sometimes they act where they're supposed not to act. And for example, DNA methyltransferases, they occasionally methylate cytosine at the wrong position, creating 3-methyl cytosine. And this would interfere with base pairing, and actually all organisms which have DNA methylation, they evolve to have this ALD enzyme which would be able to remove to actually oxidize methyl group, and this would give us formaldehyde in non-enzymatic fashion. So these guys coexist, and this is Peter Sarkia's work. He is in the Department of Biochemistry here in Oxford, and it's very, very elegant because he just looked what enzymes are present in those organisms which have DNA methyltransferases, and that's how he discovered this repair pathway and this unusual modification which can be installed by DNA methyltransferase. Another very interesting story actually is from Thomas Carell group, showing that 10 enzymes, they can occasionally hydroxylate thymine. So thymine has got 5' methyl group, and occasionally hydroxymethyl uracil. And this is as well very, very unusual base, which is removed by SMAT family of DNA glycosylase. And I'll focus a little bit for the rest of my talk on some differences in DNA chemistry and challenges which happen when templates can have these epigenetic modifications and how it's being replicated. So one interesting reaction which can happen actually naturally with cytosine is what is called the spontaneous deamination. Essentially, reaction with water, which would result loss of the amino group. So that from cytosine produces uracil. But this you don't need anything actually just aqueous media for this to happen. It's relatively slow reaction that happens. So this is the problem. The presence of this reaction is the reason why we have findings in the DNA, so we can distinguish the deamination of cytosine from potential natural bits. And so Thomas Lindholt, he observed when he looked at the deamination rates, and he repeats in here nucleotides, deoxycytidine monophosphate and methyl deoxycytidine monophosphate. We observed that actually in methylated cytidine monophosphate deaminase at higher rate than unmodified cytosine. And this was interesting observation because it helped understand why sometimes you observe that places which have methyl cytosine accumulate more mutation. So CPG mutations in CPGs actually it's a problem. It's one of the major mutational mechanisms which is observed in cancer. So for example, if we take TP53 gene, the most commonly mutated gene in cancer, 7 out of 8 commonly mutated residues in TP53 gene are CPG mutations that methylated CPG sites. So if we look at another germline disorder, where the spontaneous germline mutations emerge in MECP2 gene, 43% of mutations of MECP2 gene are CPG mutations in methylated CPG sites. And if MECP2 gene gets mutated, people develop very severe neurological disorder called X syndrome. And with kind of modern sequencing of tens of thousands of tumor samples and grouping mutations in what's called mutational signatures, so I'll just briefly introduce this type of work because I'll show a couple of those later. So on the top you see base changes. So this is base change which happen, and then each group within specific base change shows different 5' and 3' nucleotides, so all the combination of 3' and 5' nucleotides. So when mutations were grouped according to base change and 2 neighboring nucleotides, one of the most pronounced signatures, what's called SBS signature 1, is the CPG mutations, and each of these peaks contain nucleotides which have Gs. The C followed by G. So these are CPGs which typically the genomes are methylated. So methyl cytosines, while playing important functions as I mentioned in gene silencing, they are a bit of act as a mutagens in the genome. And what we're interested to study initially is to look how CPG mutational patterns change in cancers with defective DNA repair. And this is quite an interesting, again, study published from Swarcher, which sequenced lots of tumor samples and tried to score the number of mutations. Is that typically, if you sequence tumors, you discover about 1 mutation per metabase. So that's the density of mutations for most of the tumors. But there are some very unusual tumors we developed. So this is logarithmic scale from 10 to 100 to 500 mutations per metabase. So it's a very, very highly mutated tumor. And actually, they typically have better prognosis. So people who have very highly mutated tumors have better prognosis, and thought process there is that they probably generate more unnatural peptides which are recognized better by the immune system. So what happens? Why these tumors develop so many mutations? They typically develop them because they have deficiencies in mismatch repair. The mismatch repair is one of the first mechanisms which try to repair any mismatches produced by DNA polymerase when they replicate. And this is actually polymerase epsilon, major replicative DNA polymerase. So these mutations are quite specific. They typically 1 to 5 point mutations which are located in the exonuclease domain of polymerase epsilon. So polymerase epsilon gives a polymerase and exonuclease domain. So if it detects mismatches, it can actually move back, redirect DNA into exonuclease domain, degrade mismatch, and start resynthesizing DNA again. So this is the first DNA repair mechanism which actually correlates linked to the polymerase. So this mutated, if you have mutation in one of these enzymes, your total mutation load in the genome elevates. But what we were very much surprised to observe is that when we looked at CPG mutations in CPGs, in patient samples which have polymerase epsilon mutated in their tumors, we observed that CPG in CPGs increases dramatically. This was unexpected because the mutational mechanism for CPG in CPGs was thought to be this elevated spontaneous deamination of methyl cytosine. And why would then polymerase epsilon mutants would develop such a high CPG mutations in CPG? So this was the question which we are very curious about. And then what you can do, we can actually group mutations around the replication origins, and then see whether they are being emerging from the leading or lagging strand of replication. And then you look in various tumor samples, which have polymerase epsilon mismatch repair deficiency, or them separately observed enrichment on the leading strand of these mutations. So these mutations emerge on the leading strands during DNA replication. So there is some connection between CTT mutations and CTGs and replication. And that's what we aim to answer. And to study mutations is complicated in cells. And the reason is that typically, if mutation happens somewhere random in a group of cells, and you take the cells extract DNA in sequence, the allele frequency of mutation would be very, very low. And the Illumina sequencer, for example, does 1 error per 1,000 base pairs. And if you're looking at error rate, which is a couple of orders of magnitude lower than that, it's essentially impossible to detect these in kind of sequencing techniques because background rate is high. So what people typically do, you can expose yourselves to mutagen, so random mutations which emerge in the genome, but then you take a single cell and you expand that single cell into population of cells so that these mutations which happen in that single cell will become clonally at very, very high abundance at that specific position. And then you can sequence and get statistics to know that this mutation is actually mutation and not the sequencing error. But this is complicated because, you know, to do this in cells, you have lots of different things happening in cells. You cannot attribute, for example, one specific mutational mechanism to the process. So what we wanted to do, actually wanted to do in a tube reaction, but in a tube, we cannot do this kind of clonal enrichment. So we have to develop methods which would enable us to sequence very, very rare mutations, which could be produced on the DNA within the tube. So what's the experiment design which we came up with is because we wanted to determine the impact of DNA methylation, that we would take plasmid DNA, we would create a gap in it, and we would be able to install methylation in the template, or have template without DNA methylation. And in a tube, we would add polymerase epsilon hollow complex made from 4 subunits. And then polymerase would initiate and replicate that little gap single-strand bit of the DNA. And then we would like to detect mutations in whether or not it's elevated and methyl cytosine are present. So we use 3 different enzymes. We use wild-type polymerase, which have this 3-6R mutation in exonuclease and is found in cancer. And we also had a mutant which have catalytic residues and exonuclease domain mutated. So these are not naturally found point mutations, but they disable capacity of polymerase epsilon to repair errors while it's replicating. So to read very, very rare methods, we used approach which was published before and adapted it for our purpose. And the way it works, we use actually single molecule barcode. So this is our target DNA, which potentially could have a mutation. So we have DNA which would contain 19 N barcodes, and we have homology is a part of it, and then we could use polymerase to replicate that. So that's 19 N barcode would be incorporated in the original molecule, and then we do linear round amplification of that molecule. And each time it's being amplified, polymerase which you use, for example, despite the fact that you have the delta polymerase, random mutations, but that mutation is present in the original strand will be clonal in all replicated daughters of that original molecule. Then we can incorporate additional barcode, then we can amplify, and then essentially when we sequence, we get enough statistics to know individual molecules, whether or not they have real mutations or not. And so what you can do, actually, you can sequence top and bottom strands. So for example, because we have bottom strand, single-stranded conformation in the tube, we could accumulate background mutations, but we can subtract them by sequencing template and daughter strand. So we looked at precision of our method. So this is done by just mixing different mutated DNAs together at different frequencies and done after sequencing and doing this first polymerase error sequencing approach, you will be able to recover approximately the expected ratio to which we introduce that DNA to mutation frequency about 1 per million, which was good for the purposes of studying periodic polymerases. So then we wanted to see how our approach works. So we took plan of exogenous polymerase, so it's colon polymerase, so it's nuclease domain deleted, and we've done first we can look at mutational landscape and compared to the published report, and we observed that we picture a very similar mutational pattern between these ATG mutations and PTA mutations. We took another high fidelity polymerase, which was not done before, and we observed quite distinct and different mutational profile. And then we can look at total mutation errors, and we observed that as expected, plan of exogenous produces more errors than calculated with enzyme. So what happens when you do this with a human polymerase with PK6R mutant? So this is mutational landscape which we see. So this is a big type of mutation and 3' and 5' neighbor of mutated base, and we observe this CPG mutations within CPG nucleotides, where PTA mutations with CP, and this actually PPG mutations with PP nucleotides. And this is actually very similar to the mutations which are observed in tumors. So these are mutation landscapes of tumor patients, which of tumors from patients which have polymerase epsilon and mismatch repair deficiency. So then there is again enrichment of CPG mutations in CG, CPA mutations in here, and to some extent PPG mutations in here. So we're able to recapitulate very similar mutational profile. So what happens to methylation? This is a question, and we just got a few slides left here. So if you use unmethylated template or methylated template, so if you introduce methyl side in the single methyl transferase, that's the first one, to observe there is increased frequency of CPT mutations in CPT. So what happens to non-CPG context? So we look at CPH context, and we observe that there is a reduced mutation frequency in CPH context, and these two will not be methylated by cis1 methyl transferase, maybe Bartels will say. And as well, we have some sites which are methylated in one context, which is non-CP methylation, and we observe that the mutation frequency is lower. So to some extent, we are not so surprised to see that methylated CPGs would be replicated with highest error rate, but what was surprising to us that actually it matters as well which context methylation it is. So methylation on its own is not as mutagenic as methylation within CPG nucleotide. We don't know what is and this is with wild type polymerase. Again, very similar profile with methylation is a little bit higher here, comparable to CPG unmethylated, but methylated CPGs replicated with lowest fidelity. So just to make sure that this is observation which is observed in cells as well, we've done a more traditional way. We introduce this polymerase epsilon mutation of embryonic stem cells using CRISPR engineering, and we observed the CPG mutations within CPGs and elevated mutation frequency at methylated sites. So how significant is polymerase errors comparison to deamination? So deamination was measured in vitro in tubes. So approximately, just to give you a sense, one round of replication by polymerase epsilon produces the same number of mutations which it would be produced by 2.5 years of spontaneous deamination. So in the replicating cells, the thing very much matters. That's mutations probably primarily generated by polymerase epsilon. Okay, so this is conclusion. So we have error signatures which we now characterized, and we first we show the polymerase error error signature can be recapitulated to what's observed in vivo. And we observed that wild-type polymerase enzyme makes approximately threefold higher number of mutations when replicated methylated CpGs in the genome. And as I mentioned in the last slide, one round of replication equivalent to 2.5 years of spontaneous deamination. But there are very few spontaneous deamination measurements yet begun, so this is something we are looking into right now. And this is acknowledgement to people who collaborated very closely with Michael, who does a lot of most experimental work, and Marketa, who's done computational work. Thank you.